Have you ever wondered about the ethical boundaries of scientific research? It's a question that has stirred debate for centuries, particularly when human cells are used without the person's knowledge or consent. This brings us to the intriguing tale of Henrietta Lacks, a woman whose cells have made an indelible impact on the scientific world, all without her knowledge or approval. The story of Henrietta Lacks and the HeLa cells is a fascinating exploration of this ethical question. Henrietta Lacks was a young African-American woman who unknowingly played a crucial role in scientific history. Born in Roanoke, Virginia, in 1920, Henrietta was a daughter of sharecroppers and a mother to 5 children. Her life was as ordinary as any other, defined by work, family, and the occasional joys and sorrows that life offers. In the early 1950s, however, Henrietta's life took a tragic turn. She was diagnosed with a severe case of cervical cancer. The pain was unbearable. But Henrietta was a fighter. She sought treatment at Johns Hopkins Hospital, one of the few hospitals in the United States that treated African-American patients at the time. It was during this treatment that doctors took a sample of Henrietta's cancerous cells without her knowledge or consent. This act, a stark violation of her patient rights, was unfortunately common practice in the era of segregation, when the rights of African-American patients were often overlooked. Yet, within this darkness, there was a glimmer of light. The cells taken from Henrietta, later known as the HeLa cells, were discovered to be remarkably durable and fast-growing. This made them an invaluable tool for scientific research. Little did Henrietta know that her cells would play a significant role in the world of science. Henrietta's cells, later named HeLa cells, had a remarkable trait. A trait so unique it set them apart from all other human cells studied in the lab until that point. What was this trait? The ability to reproduce indefinitely under laboratory conditions? That's right, indefinitely. You see, in a typical scenario, human cells cultured in a lab would only divide a certain number of times before they'd stop. This is due to a phenomenon known as the Hayflick limit, named after Leonard Hayflick, who discovered it in the early 60s. But Henrietta's cells, they broke all the rules. They didn't stop. They kept on dividing over and over again, seemingly without an endpoint. Now you may be wondering, what's the big deal? Why is this so important? Well, let me tell you, before the discovery of HeLa cells, the transient nature of human cells in culture posed a significant challenge for researchers. It was like trying to study a comet that only appears once every 76 years. You have a very limited window of opportunity before it's gone, but with HeLa cells, that window was thrown wide open. Suddenly, researchers had a consistent, reliable source of human cells they could use to conduct their studies. It was like having an endless supply of comets at their disposal. The implications of this discovery were massive. It opened up new avenues in fields like virology, genetics, and even cancer research. Scientists could now observe how human cells respond to new drugs, radiation, and various other stimuli, all in real time. They could watch the effects unfold hour after hour, day after day, without worrying about their cells dying off. The HeLa cells, through their ceaseless reproduction, became a workhorse in the world of medical research. From developing the polio vaccine to unlocking the secrets of our DNA, these cells have been instrumental in countless scientific achievements. And so, the HeLa cells became a vital tool for researching numerous diseases and conditions. They continue to be used in labs around the world, contributing to our understanding of life at its most fundamental level. Even after her death, Henrietta Lacks continues to contribute to science. Her legacy, the HeLa cells, has become a cornerstone of modern medical research, opening avenues for scientific breakthroughs that were unimaginable before their discovery. Let's take a moment to appreciate how these cells have revolutionized science. For starters, they played a significant role in the development of the polio vaccine in the early 1950s. Jonas Salk, the man behind the vaccine, used HeLa cells to test his theories, leading to one of the greatest medical achievements in history. Today, polio is nearly eradicated worldwide, and we owe a part of that success to Henrietta and her immortal cells. But the impact doesn't stop there. The HeLa cells have also been instrumental in cancer research. They've helped scientists understand how cancer cells grow and divide, leading to breakthroughs in treatment and even prevention strategies. They've been exposed to radiation, sent into space, and have been a part of countless experiments, all in the pursuit of understanding the inner workings of our cells. And let's not forget about gene mapping. HeLa cells were the first human cells to be successfully cloned in the lab, and they've been used extensively in the Human Genome Project, a massive international effort to map out all the genes in the human body. This work has led to a deeper understanding of genetic disorders and has paved the way for personalized medicine. However, these scientific triumphs don't come without ethical questions. The use of HeLa cells without Henrietta's or her family's consent continues to spark debate about the rights of individuals in scientific research. It's a complex issue, one that forces us to balance the pursuit of knowledge with respect for human dignity. The story of Henrietta Lacks and the HeLa cells is a poignant reminder of the ethical complexities of scientific research, even as it continues to shape our understanding of human biology. Henrietta may be gone, but her cells live on, a testament to her unwitting but enduring contribution to science.